Our research project is human cloning, done by Josh Reines, Camille D'Hondt, and Dustin Blair.
There are 3 different types of cloning: therapeutic cloning, DNA cloning, and reproductive cloning.
Reproductive cloning is the most common type of cloning. First, remove the nucleus from a desired egg cell. Then, remove the nucleus from a somatic body cell with preferred genes. After, fuse nucleus with denucleated egg cell. Now you see the cloned and growing egg embryo. Finally, it is ready to insert into the uterus for a mother to birth.
Therapeutic cloning is very similar to reproductive cloning. First, remove the nucleus from a desired egg cell, then remove the nucleus from a somatic body cell with preferred genes. After, fuse nucleus with a denucleated egg cell. Now you see the cloned and growing egg embryo. Destroy embryo to harvest stem cells.
For the process of DNA cloning, isolate the DNA fragments from a chromosomal DNA with restriction enzymes. PCR, the polymerase chain reaction, is a commonly used method. Link to primary DNA sequence, and then the restriction enzyme cuts a self-replicating DNA strand. Isolated DNA fragment inserted by the ligation procedure.
Although cloning may seem like a grand idea, there are many risks. There is currently 3 to 5% success rate. Many of the clones are stillborn, meaning they are born dead. Gigantism is a common factor among clones. Gigantism is when the organs inside the offspring are too big for the body and have matured too quickly. Many other serious conditions, such as dementia, Alzheimer's, arthritis, and premature aging, are all possible outcomes of the offspring.
Dolly the sheep was the first successful clone out of 276. Dolly had been born with gigantism, arthritis, and many other conditions. The one that finally killed Dolly when she was just 6 years old was a lung disease.
Cloning has many benefits. It's up to you to decide if they outweigh the risk. Cloned organs have been tested in pigs, and hopefully will be able to transfer to patients who need organ transplants, such as kidneys. Families also want to use cloning on their pets. If a pet dies, they want to replicate the previous animal they had. Although it may look the same, the characteristics will be different depending on their surroundings. Gene therapy would be a good way to cure diseases. When people have defective genes, they would be introduced to normal genes, which will counteract the disease and teach the gene to behave normally. This is hoped to help with serious diseases such as AIDS.